Genetic modification. It has been mainstream controversy since the late 1990s, started raising red flags in the 1970s, and occurred since agrarian human history. Thousands of years ago, humans began great genetic experiments. Plants and animals were domesticated, producing many varieties of the same species. Maize, potato tubers, brassica, bovine, equines, swine, and the list continues. Domestication is a process in which organisms are bred to be tame, live in close association with humans, and assist in labor activities. Domestication dates as far back as 15,000 years for animals and 10,000 years for plants. As far as animals are concerned, there are 6 criteria that make for a candidate for domestication, which can be used as a model to explain why not all animals are domesticatable. These criteria include being a generalist in terms of diet, reach adulthood relatively quickly, be capable of being bred in captivity, have a calm temperament, not be startled easily nor flee when scared, and conform to a social hierarchy. Through trial and error, aka the scientific method, our ancestors carried out genetic experiments to create new species or subspecies that are docile, submissive, and have exaggerated characteristics. Take, for example, domesticated cattle, where 5 species of wild cattle have been domesticated approximately in the last 10,500 years. Through the domestication of these species, humans had a reliable source of milk, hides, meat, and the capacity to carry out laborious tasks such as plowing fields. Every cause has an effect, and each effect in turn becomes a cause itself for a future effect. Agriculture and domestication of animals created 2 basic types of lifestyle: settled farmers tending their crops and livestock in the richer farmlands, and nomads wandering with their herds of sheep, goats, and horses across the dry grasslands on the fringes of civilization. Domestication was one cause, one catalyst for urbanization. Urbanization then caused for more ease of trade and communication, but also the spread of infectious disease. Urbanization and improved agricultural practices also caused for increased food supplies, which then caused an increase in the population size. This chain reaction of events can be followed through modern times. One pathway we could follow is with infectious disease and the development and transmission of antibiotic-resistant bacteria. This is something that I doubt our ancestors considered as a risk while they genetically modified our domestic crops and livestock. However, they would have had to anticipate what would happen some 10,000 years after their lifetimes. Focusing on present day, humans are still completing great genetic experiments, but by using much more sophisticated techniques, such as gene transfer. Gene transfer produces transgenic organisms, where it contains a gene to produce a desired protein product, and where this protein product is not native to that organism. The technique has produced E. coli that can synthesize human insulin, and agricultural crops that can produce insecticides, which limits the amount of chemicals needed to be sprayed on our food sources. This process begins with identifying a gene of interest that you wish to transfer. It can be accomplished one of 2 ways. The first is to cut out the gene of interest from DNA using a restriction enzyme or endonuclease. The other is to produce DNA from mRNA using reverse transcriptase. Once the segment of DNA, or gene, has been isolated, it can be amplified using PCR. Then a bacterial plasmid is cut open using the same endonuclease as in the first step. The blunt or sticky ends of both the gene and plasmid complement each other, allowing them to be spliced together by DNA ligase. The recombinant plasmids are cloned, inserted into new host cells by shooting, spraying, or heat treatment. Scientists need to assess the risks associated with the creation of transgenic organisms. This is accomplished through the application of the precautionary principle. This principle states to first do no harm. If the risks outweigh the benefits, research or other activities should be discontinued. If the benefits outweigh the risks, research or other activities could continue. In defining risks, they are those that cause physical, psychological, social, and economic harm. In class, we will investigate and analyze the risks and benefits of gene transfer using Bt crops as a model. Bt crops contain a gene from the bacterium Bacillus thuringiensis. When this gene is expressed, it produces a protein that is a highly effective insecticide. When the gene is transferred to plants, they produce the toxin in every part of the plant, including the leaves, roots, stems, flowers, pollen, and fruit. These crops were developed by none other than Monsanto. Consequently, the Bt gene and others, along with the techniques for producing transgenic seeds commercially, have been patented in the United States. Based on this information, what are some risks you can anticipate from Bt crops?